Hello, everybody. Welcome back. So we are continuing our discussion of the cell cycle. In our previous lecture, we were really looking at where a cell spends most of its life, that being in the interphase stage. And now we're going to be focusing specifically on mitosis and cytokinesis and all the phases that are sort of happening within this important time of active cell division. So the main thing that's important to know is that this is occurring again in a series of phases, and we call this sort of the M phase for mitosis. So there's a prophase, a metaphase, anaphase, and a telophase. And before that, we have what we talked about previously, the interphase. Easy way to remember it for me is interpro, meta, ana on the telo. So your book has a really great graphic looking at all the different phases, and so I thought I'd highlight each of them here because they really are some pretty incredible graphics, and it's really looking at actual microscopy images of both animal and plant cells at these different phases. So you can see here in animal cells, again, this preparatory phase, we're basically replicating, again, all of our genetic material so that we have two sister chromatids connected by that really important centromere. And then the other thing that's visually that you can see here in these slides is that the chromosomes are loosely gathered around the nucleus, or in the nucleus. But when we start to move into prophase, we start to see something a little bit different. We start to see that these chromosomes that were, again, now we have that phase where we have two sister chromatids, they're getting bunched up. And you can see how compared to the previous slide, the chromosomes are much more bundled together. And importantly, the nuclear membrane starts to disassemble so that basically the sister chromatin can start to move around the cell. And the last thing that's really important is this microtubule fibers start to form the mitotic spindle. And that's going to be a really important part for the next couple of steps. So really important that we form this mitotic spindle, because this is really where all of your chromosomes are going to attach and ensure that your cell divides efficiently. So then we move on to metaphase. An easy way to remember this is M for middle. You start to have your microtubule fibers from opposite ends attached to those chromatid, and really attachment to that spindle apparatus, that middle part, these cartilaginous tubes that allow for the centromeres to attach and create this very distinct middle alignment of all the chromosomes. So you get the chromosomes that are basically easily centered in the middle of the cells. And then you have anaphase, and this is where those microtubules start to shorten, and it pulls apart those sister chromatids to opposite ends of the cell. So where before we only had one, where we had these two sister chromatids, they're being pulled apart to opposite ends of the cell, and this is anaphase. The next phase is telophase, and this is basically really showing that we're getting all the chromosomes on each side of those poles. You start to get that really important spindle fiber to disassemble, and importantly, nuclear membrane starts to form around each of the chromosomes, so creating two daughter nuclei in what is going to be each of these new cells. And then lastly, we have cytokinesis. And this is what we talked about previously, where basically In animal cells, the cell membrane pinches into two separate daughter cells, and in plant cells, a new cell wall starts to be built between the two daughter cells. So a little bit different mechanism between plant and animal cells. Sorry, there was a little bit of a glitch with Kaltura, the photo editing, the video editing software. So where I wanted to spend a little bit of time is on this concept of the mitotic spindle. So you might not have really looked specifically at mitotic spindle development in your other biology classes, but really it's one of the most important processes in cell division is the construction of this mitotic spindle and the successful migration in anaphase and the separation into the two daughter cells. And the thing that's the mechanism that's really the most important about this mitotic spindle is it's the structure that separates the sister chromatids during mitosis. So the centromere attaches to the spindle, and then then they are physically basically torn apart into their sister chromatid structures. And this mitotic spindle is made out of microtubules, and that's really just these hollow protein fibers that again assist in the building of this mitotic spindle. And as you probably remember from our previous discussions about important cellular organelles, we talked about how these microtubules are a really important component of the cytoskeleton. And so another important feature of the spindle is I mentioned that the centromere attaches into the chromatids onto the mitotic spindle via the centromere. And the important thing is the binding that occurs there are on something called kinetochore proteins. And that's what enables the centromeres to bind to that spindle. And ultimately, that's really what is driving the system to allow mitosis to successfully divide these cells. So we'll pick up in our next lecture about understanding the concept of cancer and cell division and what can go wrong in cell division. So now that we have a good structure and understanding of the processes that are involved, we're going to spend some time understanding what can go wrong with cell division and how that can potentially manifest as cancer in the body. Okay, see you next time.